guys do, and I never leave the cerebellum without pointing this out. These are arguably some of the second most famous neurons, if you will, easily recognizable. The lower motor neurons, I know that. Pyramidal neurons in the cortex, I can see that immediately. And these guys, number 2 or number 3 on the list would be the Purkinje cell layer of the cerebellum. You see how big they are? These neurons, you see how large they are and how they're collected in this layer right here? These neurons really emphasize the basic functional role of cerebellum. So we know that the cerebellum has some subconscious role, stuff that it plays for the human body. We also know that it influences skeletal muscle contraction. But the major thing about the cerebellum is its modulation of systems that exist elsewhere. So in other words, the cerebellum has inputs to systems that are already running. And it appears that the systems that are running already are all overstimulatory. That is, they need to be slowed down. And so that appears to be the major function of the cerebellum, and these Purkinje cells illustrate that. They are down regulators, the Purkinje cells are. They release a very famous chemical called GABA that slows things down. If it helps you to remember the slowing down effect of the Purkinje neurons, if it helps you to think about it, the drug Valium, everybody knows this drug is a relaxer in the nervous system. It's an inhibitor. It binds to the same receptors, the benzodiazepine receptors, that GABA does, that are produced by the Purkinje cells. And so you think about the Purkinje cell layer as a Valium, a natural Valium, slowing things down, and it speaks to the general function of the cerebellum. Okay, so I've shown you really famous neurons, lower motor neurons, cerebral cortex neurons, Purkinje cell neurons of the cerebellum. That was bonus. All right, to the skin. And then a few more introductory things, a couple more class periods, and then we're going to do a review. It'll be time for a test. Yeah? So it's coming on now, right? Just a couple of weeks away. So let's review first what we already know about skin. So I taught you epithelium already, did I not? And I said that epithelium covers in lines. On the outside it covers, and on the inside it covers. And we looked at 2 varieties of stratified squamous epithelium. I said stratified squamous epithelium can be keratinized or non-keratinized. You see, we describe the tissue based on the presence of a protein. This time the presence of keratin, or the absence of keratin, describes it. So generally speaking, the soft stratified squamous linings of our nose and our esophagus, anus, places where friction, where we lose a lot of cells, that's stratified squamous characteristic. In those places, they're non-keratinized, soft. The outer layer of our body, epidermis, is keratinized. So fingernails are keratin. That is a different variety of keratin that makes this part of the epidermis, the fingernail. And the rest of our skin that covers us is stratified squamous. Now, sorry about that, but you're going to have to learn the layers of the epidermis. I'm going to teach you those today. But first, introduction. You can see the epidermis here is stratified squamous. Now, also notice, as we work our way down through the stratified squamous epithelium, that the thickness of the skin, the bulk of the thickness of the skin, is in fact not epidermal. It is dermal. And this dermis here, we looked at a histology section of as well. It didn't have all the rest of this in it. But the dermis is dense, irregular connective tissue. We described dermis. We described the joint capsules that we look at under a microscope in relationship to the arrangement of the collagen there. So it's an irregular arrangement of collagen. So stratified squamous outside, dense irregular connective tissue for dermis. Now, once we have learned the cells, most of them are keratinocytes, and said a few things about the layers of the epidermis, Then we're going to spend the rest of our time talking about the stuff that we see in the dermis. In your textbook, the textbook is not going to go into the details here that I am. Okay, you have to go to other chapters in the book to pick these up. But now is the time to introduce young people to the stuff in the dermis, because it opens the door for other things that we have to learn in anatomy. For example, if I say to you that within the dermis we're going to have several different varieties. You can see a Pacinian corpuscle here, a Meissner's corpuscle here, a free nerve ending here. We have several different varieties of sensory capacity within the dermis. Doesn't it make sense now to build the outline of that? Let's make the outline in our minds right now, so as we get to it later, I can fill in the details. So I'm going to take a Pacinian corpuscle, get on board Miss Frizzle's school bus, and take that information from the Pacinian corpuscle back to the brain, and I'm going to name that pathway. So you're going to have learned, by the time we get through the introductory material, what a pyramidal motor tract is. Have you heard that today? And you're going to learn what a spinothalamic sensory tract is. Where is that information? So that I can begin to fill in those details as the semester wears on. All right, there's also some other things here that I've mentioned to you. There are erector pili muscles. And students look at this erector pili muscle attached to the hair follicle and they go, you know, that looks a lot like a skeletal muscle, doesn't it? But a good anatomy student who's had a teacher to point these out to them knows that's not skeletal, is it? No. What kind of muscle is that?
Smooth.
That's a smooth muscle. That's right. And this one, it looks like skeletal. It behaves a lot like skeletal in terms of its regulation. The varicosities are interlaced within it. So I have more specific control than I would for a sheet of muscle cells like in the GI tract. So this guy right here is going to function more like a skeletal muscle. But its information, making it contract, is going to come from the autonomic nervous system, the involuntary nervous system. So when we talk about skeletal muscles, we're going to track pathways there that are voluntary. When we go to smooth and cardiac muscles, we're going to go involuntary tracks. And so this will help me bring this up again, talking about the erector pili muscles. And that picture is going to show up again before we're done with the single and multi-unit smooth muscles. So bring up the nervous system, bring up the smooth muscle stuff again, bring up the connective tissue types. All right, the other thing you see here is vascularization. So I pointed this out. I said, y'all, epithelial tissues are defined based on the cells there. Flat cube column. One layer, multiple layers. And epithelial tissues, we know, do not have blood vessels. They're avascular. However, they are innervated. So let's look for it, shall we? Let's look for innervation in the epithelial lining of the skin. Let's look for the fact that there are no blood vessels there. And then let's remind ourselves that the underlying connective tissues are going to be the source of living material for the epidermal cells. And that stuff is going to come from the underlying blood vessels. And you can see them as they innervate into the papillae, the little finger-like projections of the dermis, your fingerprints that exist here pressing into the epidermal layers, bringing nutrient so that it can diffuse through the basement membrane into those epithelial cells. All right, and then we're going to talk about, just bring them all up here, then we're going to talk about the glands here. As it turns out, there's a bunch of different varieties of glands that we will see. For example, this one here labels, can you see the light purple here? You can see it there, extending the duct, extending up there. That's the most common type of sweat gland we have in the skin. It's called an eccrine sweat gland. And then I'll talk about apocrines with those. And then there are these really interesting glands that are always associated with the hair follicles. Apocrines are like that. but also these oily secreting glands called sebaceous glands. They make a product called sebum that is the main problem for acne, because these are sensitive to hormones and they overproduce, and when they do, they clog the pore passing up through the hair follicle for the skin, creates a nice, warm, moist environment there where bacteria like to live. And so that's the cause. Every drug, Accutane, all of the soaps, etc., that are built to treat acne deal with that gland right there, to try to dry those glands, to try to clean them enough, keep them clean enough so that they can get the sebum through the pore, through the hair follicle pore. All right, and then the final thing here before we start it, this is all just introduction, is the hypodermis, the underlying, sometimes this is referred to as the superficial fascia of the human body. The hypodermis, the adipose tissue, which we have also talked about already, that covers the entire body, the superficial fascia. And hormones can regulate this, the distribution of this as well, and we mentioned that just very briefly. Most of this we pointed out, some of this we haven't, and for those, we'll point those out as we go. All right, first, the cells of the epidermis. 4 cell types. The most abundant type is called the keratinocyte. Now, why would I call the cells of the epidermis keratinocytes? More than 80% of the cells there are keratinocytes. You know, there are 2 kinds of stratified squamous. There is the non-keratinized and the keratinized version. These are cells that contain keratin. I'm going to say just a little bit more about that on the next picture. The second cell type that I do want to emphasize a little bit to you in terms of its location and its importance is the melanocyte. Melanocytes reside, the cell bodies of melanocytes reside in the lowest portion, the deepest portion of the epidermis. They sit right on the basement membrane. And these are the primary producers of skin color. Now, you're going to learn that there are 3 really powerful skin pigment producing structures, chemicals. This one is by far and above the most important for color, the melanocytes that exist here. So when we look at a section of skin, we're going to look for melanocytes. It is the melanocyte production combined with the melanocyte degradation, or the melanin degradation, that describes it is the number 1 describer of why humans have different skin tones. How many melanocytes are there? That one's not always a good predictor because most human skin has the same number of melanocytes in it. Can't really tell with that. But it's the production, it's the type of melanin produced and how the skin handles that melanin. Now, that's a physiology thing. I put 1 biochemistry slide in this presentation. I'm not going to make you learn it, but I want you to see it. This describes skin color. Melanocytes are also the most dangerous skin cancer. So I'm going to point that out to you as well. That's why I told you these cell bodies sit on the basement membrane. And so they can very quickly metastasize. They can create clusters and create melanomas that become it's like the number 4 most dangerous cancer in the human population, melanoma. Melanoma is a very bad diagnosis and very poor treatment outcomes for a melanocyte that has broken through the basement membrane and started to spread. So melanocytes are important. We'll point them out. The third group, the Langerhans cells, are epidermal macrophages. These are cells of the immune system that reside in the skin. I went and had an allergy test done, and they took these little needles and they pricked my skin with them. And then they could tell whether I was allergic to it or not because I got a little rash, a little red bumps on my skin where that little needle prick was. How did the immune system respond to that? Okay, well, there's several ways, but this is one of them. There are macrophages here called Langerhans cells that can activate the immune system. And the way macrophages do that is fantastic, the way they do it. They take the thing and they show it to the helper T cells. So the immune system is here in the epidermis. The final cell type, some argument as to how important these guys are, but you got to show them. The Merkel cells, because they're there, they're believed to function as touch receptors. Now, it is more appropriate, y'all, and I'm going to emphasize this. It is more appropriate to think about touch in the skin as a phenomenon of the dermis. There is in fact free nerve endings in the epidermis as well. Right? It's innervated. Right? Avascular and innervated. But the touch stuff that I'm going to emphasize to you are the locations of the encapsulated and non-encapsulated pieces of the nervous system in the dermis, even with your hair follicle. So the most sensitive touch that you have on your skin is when somebody touches the tip of a hair. You don't even have to touch your skin. You can just touch the top of your hair, and you can detect that. That's because that epidermal derivative protrudes down into the dermis. And as it does, as it protrudes down into the dermis, you can see that there are free nerve endings that surround the bulb of the hair. And so any movement of that hair at all is going to stimulate those free nerve endings of the hair bulb. And you will be able to detect that. So, like I said, we're going to do most of the nervous system stuff as we proceed down into the dermis. All right, those are the cell types. Here are the layers. The major layers of skin are outer layer, stratum corneum. Second layer, stratum granulosum. Third layer, stratum spinosum. Now, thick skin has another layer here called the lucidum, but it's not on the typical slide. It's present mostly on, the soles of your hand, palms of your hands, soles of your feet, top of your head. It's not as typical, but anywhere you go, you will see these four. Thick skin has a fifth, but thin skin has corneum, granulosum, spinosum, and basale. Now, the stratum corneum is the outer layer, 20 or 30 cell layer thick, dead cells filled with keratin. So as this says here, the cells are dead, and they're just membranous sacs filled with the protein keratin. This is the outer layer of the skin. It's the protective layer. It's replaced every 30 days. Skin cells by the thousands upon thousands are lost every day from the stratum corneum. The next layer down is the granulosum, and it also looks like multiple layers of flat cells. Now, the granulosum has some characteristics to it, and I'm not going to ask you to remember all of the lamellated granule story and the differences in the keratin. But what I want you to realize here is the name. If you know the name, it will help. If you think about the name, it'll help you remember where it is. Okay? So in anatomy, when you describe where cells came from, you will oftentimes refer to those original locations as granular locations. It could be that you want to call it granular because the cytoplasms are filled with granules. But in the original description of the granulosum, and we see this in other places, in the original description of the granulosum, the anatomists believed that the cells that were dying and being sloughed off on the corneum were actually being produced in the underlying granulosum. We know that's not true now. These cells of the granulosum came from the same place the corneum cells came from. They came from this basement layer, from this basal layer. So all the cells, this is my chance to say it, all of the cells of the epidermis come from the basal layer. Cell division is occurring down here, and then those cells are being pushed outward. So all the cells of the granulosum and corneum originate in the basal layer, and then they push outward on the epidermis as more and more produced. The third layer is spinosum, and you already know something special about spinosum. I promise to point this out to you once again. The spinosum. There's something special about the spinosum. Do you remember? Something special about it. Skin's pretty tough, isn't it? Is the corneum going to help with the structural integrity of the skin. What happens when you scratch the corneum? It comes off. Yeah? So the first explanation for integrity, for structural integrity of skin, is here in the spinosum. Because these cells of the spinosum are connected by desmosomes, like rivets in a boat. There is strength in the connections of the stratum spinosum. It's the first explanation for skin integrity, the stratum spinosum. Now, I'm going to mention it with one more context for you as we're passing through the skin cancer descriptions. There are three kinds of skin cancer that we'll talk about, and one of them originates here within the stratum spinosum. The other two originate down here. Okay. Now you can pick up the Langerhans cell here, most of the time located in the spinosum as well. Okay, now the last layer of the skin is the stratum basale, and you can see several important things here in this layer. First of all, you can see the presence of a melanocyte. Now I want to point out something anatomically about this melanocyte for you. I want you to notice, even in this cartoon depiction, that the cell body of the melanocyte here is intended to be visualized here as having a finger-like extension that extends to there, and there, and there, and there. Can you see where those gray things are ending there? Can y'all see that? That's on purpose here, because the melanocyte cell body does sit on the bottom layer of the epidermis, but it has finger-like projections that lace through the spinosum. And so the melanin granules that are produced in the melanocyte migrate through those fingers, through the spinosum cells, and then make their way into the spinosum cells, as you can see here. Can y'all see the dark black pieces there? Some of these are intended to illustrate desmosomes. These here are intended to illustrate melanin granules. Can you see where they're orienting? They're all orienting on the external or superficial portion of the spinosum cell. That is a known fact. The melanin granules that are released from the melanocyte orient in the spinosum cells on the superficial portion, creating shading for the nucleus. So when your skin is exposed to damaging sunlight or damaging tanning bed scenarios, and the melanin is produced in greater concentrations, the melanin itself tries to prevent damage to the nuclear material of the stratum spinosum. It is a shading technique for the genetic material that's in the nucleus of those cells. I find stuff like that fascinating. So the melanin here you can see from the melanocyte. And then finally, here's the Merkel cell or the touch cell in the stratum basale. Okay, so there are your layers. A few observations about each. A mention of the change in the keratin, finally terminating with the corneum being nothing more than sacs filled with that protein. Those are dead cells. All right. That's a great stopping point right there. I'm going to pick up with the dermis. I've done epidermis. Now, I should say this. I am going to say some more about color. But I put those things in categories. So we'll return to the epidermis to talk about color again. But I want to do dermal anatomy with you as we begin class next time.